Hello everyone. We have been talking about a polyphasic approach in bacterial taxonomy, right? So in previous few lectures, we have talked about what the polyphasic does mean, what are the different techniques that we are discussing about, out of which G plus C contain TM value, DNA hybridization, etc. We have been discussing, right? So when it comes to DNA hybridization, short form is DDH, is a technique, as we know, that measures the similarity between DNA sequences. May you have to check for the similarity between the sequences which are obtained from different organisms. Yes, if you can see in the diagram here, you are seeing our ancestors, right? Chimpanzees and gorillas and orangutan and the Homo sapiens sapien. Now, why I have shared this? You can see in the slide also on the left hand side, you have the DNA hybridization matching uh, that is shown, right? So you have DNA sequence fragment from flies, the fragments from human, and that is from chimpanzees. So as you can very well say that the DNA hybridization studies, when it is done between the DNA isolated from a fly and that is from the human cell, do not match, right? There is no pairing as such, which is shown by the bulging areas, right? There are no hydrogen bond forms between the complementary strands. But as against that, when it comes to hybridization study between human and chimpanzees, very, very it is clear that yes, maximally most of the uh, sequences are found to be complementary to each other, and there are hardly any one or two bases in this particular series of say 15 bases is found to be not complementary, right? What does it mean? Chimpanzees, at slightly more than 1% of their DNA sequences are similar to human, and 2% between human and gorilla. So this is the degree of similarity of sequences, right? So as we know, DNA hybridization is a measure of degree of similarity between DNA sequences, and that is done by hybridizing labeled DNA from one organism and the unlabeled DNA from another organism. And the degree of hybridization is determined by measuring two factors that we have seen: thermal stability of the hybrids, that is reassociation kinetics, right? That is what we have already discussed in previous lectures. Now, when it is about DNA hybridization, of course, it remained as a gold standard earlier, the ultimate method for finding out the genomic level classification of organisms. However, with advancements in the techniques, uh, it was found to be like there is a necessity to replace this DNA hybridization, or maybe it can be said as to complement this method. It alone cannot give you the required identity, right, of the particular organism. It cannot be properly placed in taxonomic units. So considering that, the time required for the process, came complexity in the chemical steps, these were a few challenges. In addition to that, when it comes to DNA hybridization, though it was followed for years together and to construct the classification system, the genomic groups, genospecies of strains which share similarities, more than 70% similarity, they were put together. But of course, the very first point is the comparative study of different methods. They indicated that value of 70% of similarity could not be used as absolute boundary for classifying and taxonomic positioning of the isolates. Right. So you have to have something which is going to give you much more important uh, or useful tool, I shall say, that would assure the similarity. So there won't be a misplacement of an isolate. Yeah. So is DDH absolute? No, of course not, because 60 to 70% similarity also is going to clear-cutly divide organisms into different taxonomic groups or genus levels classification, it can be very well achieved. So DNA hybridization, yes, it has its own place, but needs to be complemented with some additional methods, right? And of course, limitation, as I said earlier, the complexity of DNA hybridization method, and it is time consuming in the nature. And when it comes to complete genome sequencing, how long you can you will be running the process, you can imagine, right? So building cumulative database based on DDH is really cumbersome or difficult and remain the major drawback in the bioinformatics era. We know how do we go now these days. We use the NGS platforms. We have talked about it. We sequence the entire genome also in few hours, and then it is the sequence is blasted and compared with the library, compared with the databases in the libraries, and you get to know that is blast, what we call it as local alignment fragment analysis that we do, and that is going to tell you about the similarities or differences and percent similarities, and you can very easily find out the taxonomic position of the organism that we all know, right? So when it comes to data obtained from DDH, it is really a limitation when it comes to the cumulative databases and building of those and utilizing those, etc. So considering these limitations, people have talked about some supplementary methods. One of this is 16SrRNA gene. Of course, it is not really new to you, but I'm going to try to summarize it so that you get to know what the technique is and how are the details. All right. So when it comes to sequence alignment or sequence similarities, what sequence is to be chosen for DNA hybridization? The entire genome or the fragment of the genome? Okay, if we go with the entire genome again, we are finding the limitations. So at the same time, can you imagine two bacterial species or strains can be divided into two as strain 1 and strain 2, or subspecies 1 and subspecies 2, etc., just by single protein coding genes, which is present in one and which is absent in one, right? So this is very important. But yes, when it comes to resolution of the methods, you need to have reassociation experiments. Surely DDH cannot be absolute 100%. Yes, it was a gold standard earlier, but needs to be supplemented with other methods. Now, what are the other methods? As I said, single protein coding gene sequence also can tell you about how do the two isolates they vary with respect to each other. Similarly, analysis of relationship based on multi-locus sequence analysis. It is also referred to as MLS method and analysis ka A. Right? So multi-locus sequence analysis, it is suggested as a primary approach for substituting DNA hybridization. Right. Of course, here is one example ki Burkholderia species, which has got lot many applications on the commercial market today. So when it was approached for classification for specific group positioning, it has major drawback that arise from a putative bias in gene selection and amplification primer availability. That is another concern when you go with locus sequence analysis, locus based, maybe single locus or multi locus, because when you are going to go with these methods, you have to have amplification, and the amplicon requires primer, and availability of primer for a new organism and a gene of interest whose functionality maybe it be discovered or sequence is to be yet discovered becomes a challenge. So these are few points to be discussed when you are talking about DDH as a gold standard and what can substitute it, and say maybe 16SrRNA. Look into 16SrRNA into details, but before that, there are other methods which are coming up to replace or contribute or complement DDH. So one of these is ANI, average nucleotide identity. So between a given pair of genomes, if you are going to calculate average nucleotide identity, right? So hybridization, say you are going to get percent identity. Here you are talking about averaging the value. So it is an alternative for gold standard. right? So pairwise genome comparison of all shared orthologous protein coding genes, that is one of the methods. So orthologous protein coding genes. So protein coding genes, that is what you are talking about, and you are comparing those genes between two organisms. So the technique is average nucleotide sequence. So sequence from those coding regions which code for the proteins, and then you are going to compare that. What happened to this? Is it really comparable to DDH? Yes, evaluation has shown that the resulting averages reflected the degree of evolutionary distances between the compared genomes, and a value of 94% identity could be represent the DDH boundary of 70%. So with the help of DNA hybridization studies, you are going to uh, put organisms into different taxonomic units based on the percent similarity, less than 70% or more than 70%. But when instead of DDS, when you explore ANI method, then you can get the identity up to 94%. So, an advance in the comparison that may better reflect the degree of reassociation between the DNA stretches of two genomes. So it was achieved by comparing artificially sectioned genomes. So you have to cut the genome into fragments, and then you have to go on reading the sequence and see the identity in both the sequences. So of course, there is another term here written as whether or not they responded to real open reading frames. So whether the gene is going to start expressing itself from at that point or not is not taken into consideration. Fine. So I need not go into these details, but at least you should remember the terminologies. Like I said earlier, multi-locus analysis. Then now it is average nucleotide identity. Then another is MUM, which is maximal unique matches. Right. So predicted protein coding sequences. So other parameter is MUM, that is maximal unique match. So like that you have metagenomic approaches which complement the genomic approaches. Right. Now come back to the point, that is rRNA homology studies. Now it is accepted that rRNA the best target for studying phylogenetic relationship. Why it is like that? Because it is said to be the most conserved regions also it has, and more divergent regions also are there in this particular sequence. Right. So you have most conserved, and you have most variable regions or variable domains they are found in. So conserved regions shows the similarity, and variable regions will reflect upon the divergence. So the concept of rRNA sequencing, when it comes to what it is, how it is utilized in the taxonomic positioning, and what is the procedure that we are going to talk about. But what is the reason, reason that it is used as a taxonomic marker is, as I said earlier, it is composed of highly conserved as well as more variable domains. That is one. In presence of almost all bacteria, often existing as a multigene family or operons, right? Then function of the 16SrRNA gene over a time has not changed, suggesting that random sequence changes are more accurate measure of time. So it will show the divergence, as I said. So with evolution, uh, evolution is to time factor, of course, with reference to time, how and what kind of variations are happening, that also can be graphed when you are going to use it as a marker. And of course, 16S rRNA gene, which is about for 1500 base pairs, is large enough for informatics purpose, which is the limitation for DDH. We talked about it, right? So uh, let's see the method in details. This was a background for the discussion. Now here is the point: characteristics of 16S rRNA. So is a ribosomal RNA necessary for synthesis of all prokaryotic proteins? We know that each bacterium contains 5 to 10 copies of this particular molecule. Then multi-information system, you can say it is composed of variable regions and conserved regions. The length of the moderate 16S rRNA is about 1500 bases, which contains about 50 functional units or domains. And the sequence encompasses the promoter region, which is very important in expression of the gene, or you can say initiating the transcription. And the core structure, when it comes to the features of nucleotide sequence, they are present in 16S rRNA, include loop arrangement of single stranded RNA and the secondary formed by hydrogen bond between bases. This is the basic information which you have learnt about 16SrRNA in earlier classes. Now let's go into the details. This is what I'm talking about. So this is your structure of ribosome schematic representation. This is the last subunit, small subunit. Last subunit has 5s rRNA and 23s rRNA, and the small unit has 16s rRNA along with the proteins. All right. So it was Carl Woese and George Fox who had used this molecule 16s rRNA sequence as a tool in phylogenetic studies. Right here, if I talk about this RNA, which is extracted from ribosomal unit. Now you can see the 16S rRNA molecule which is folded into loops and arms. So this is the schematic diagram that shows the architecture of this molecule. However, below that you can see some a strand which is showing you some bands which are marked in red and some bands which are marked in green, which are going to tell you about the red is to stop and green is to express or is to be to indicate start or proceed. Right now, what is the connection here? Let me talk about in the next slide. Here I am showing you the uh, same diagram. Along with, you have certain regions which are marked in light green. Right, so it is labeled as v1 to v3, then v3 v4 v5 v3 v5, right, and V4, V6 to V9. So overall, the full length, if you scan, it is about V1 to V9, right? So this is what is the entire variable region of the 16S RNA, right? So any of these can be used as a marker for bacterial classification. So what you have to remember, 16S RNA, when we say, it is not that, but you have to remember that it is uh, been you can say described in terms of variable regions and conserved regions. Variable domains, they are v1 to v9, right, and the respective dimensions also are given in terms of base pair regions, and their position minus indicates upstream the promoter. So and approximate number of base pairs for particular sequence also are given. For example, v1 to v3 region is about 510 bases, right? So we need to know these are the sequences which are to be used as the uh, regions which can be compared when either you are comparing the divergence variations or when it is about comparing the conservation, relative conservation, right? So red and green areas that you have to see, the red zones, so v2 and v8, for example, very brightly they are seen as red. They tend to exhibit low resolution at phylum level. So what is their importance when it comes to comparison, sequence comparison? That is what we are doing in 16s rRNA, right? So how far they are useful as a tool when it comes to phylum level resolution. So the red zones are comparatively going to be less important, or you can say lower resolution at phylum level. The green zones v4 v5 v6, they correlate with the shortest geodesic distances, suggesting that they may be superior choices for phylogenetic analysis. Now how do we do this analysis? That is the next step. Let's see here, 16S rRNA sequencing processes, right? So you have DNA extraction that is to be done, then DNA to be extracted. Now when I say DNA extraction, PCR amplification, library construction, sequencing and data analysis, these are the major steps. But when it comes to DNA extraction, and you are talking about 16S rRNA sequencing, so how to start? Of course, in the genome you have regions which encode 16S rRNA, isn't it? So that 16S rRNA coding gene is to be extracted. So of course, in some of the references you will find people have utilized 16S rRNA sequencing, and some of them they talk about 16S rDNA sequencing, राइट। आरएनए का सीक्वेंसिंग अलग एंड 16s आर डीएनए का सीक्वेंसिंग अलग. So whether you are sequencing the RNA molecule or whether you are sequencing the DNA of that DNA, or you can say the gene of that uh, segment, that is gene which encodes 16s rRNA, accordingly the term will be varying. But overall it is DNA extraction, then PCR amplification, library construction, sequencing and data analysis. We have to talk about these one after the other, but in the slide you also see one more factor, that is the different platforms that are used for sequencing the regions of 16s rRNA. As we have seen, V1 to V9, right? So the Illumina MI6 is a CQ, that is for sequencing, so which is good for commonly utilized for V3 and V4. When it comes to V1, V3, V3, V5, and V6 and V9, that time you have Roche, that is another brand, or you can say tool with the name Roche, which is going to be utilized for sequencing, the sequencing platform, NGS sequencing platforms, Roche 454. We have talked about this also. Illumina HiSeq, this also we have talked about, which is commonly used for V4 segment or V4 domain of 16s rRNA sequence. And another one you have Pacific Bioscience, that is the name given to it. Of course, it is brand name. We know V1 to V9, full length about 1500 base pair sequencing. So for sequencing, you have so many different types of platform available, and selectivity is based on the base pair size, that is one, and specific platforms companies have developed with reference to the efficiency of the process. All right. But it is just to say in one sentence that the variable domain regions in the 16s rRNA sequence are sequenced with the help of NGS methods, which include like Illumina. Right. Now, how does it work, and what are the uh, individual steps that we have to see? Right. So this is your 16s RNA, right? Ribosome. Here is the 30s unit. This is 50s unit. We have seen it in the previous regions also. So this is another way that you can represent schematically the 16s rRNA, which is of importance in phylogenetic classification. Right. Now with this, I'm going to stop sharing and going to talk about the points that are enlisted till now. All right. So what I have talked about till now is uh, सिंगल लोकस, सिलेक्टिव लोकस विच एनकोड्स फॉर पर्टिकुलर प्रोटीन. जैसे कि अभी और यू आर टेकिंग अ सिंगल लोकस विच इज 16s rRNA सीक्वेंस. राइट नाउ ये जो है, why this, that will come to come to talk about it in the end. But just now let's discuss about the methodology as such, just to revise. All the points are known to you, how is DNA extraction done, how is PCR amplification done, how is library constructed, and all that. But just to revise in a short go, we will talk about it. Right. So when it comes to workflow of the process, the key steps, as I said, genomic DNA extraction, then targeted PCR amplification. When I say targeted, you need the primers, then you have to construct the library, sequence it with the NGS platforms, and go for bioinformatics analysis. When you talk about sample collection, you have to be very careful, right? So you don't want any contamination in the sample. Sample integrity is very important. You have to extract the DNA, and you have to go for PCR amplification, targeting specific regions of 16s rRNA gene, right? And you have to use gene specific primers. So 16s rRNA gene, when you say, you have to use gene specific primer. And further, it is about different domains. So are you going to uh, screen the entire 16s rRNA, that is for 1500 base pairs? That is one approach. Maybe once it is done and still you are not able to classify phylogenetic positioning of the bacterial isolate is not complete, then you might have to go to restricted zones, which are V1 to V9. Right. And the importance of this V1 to V9 is very well described. We find research papers based on that, but I'm not going to uh, uh, um, talk about it right now. But to say yes, maybe total entire 1500 base pair sequence, gene sequence of rRNA, you have to amplify using the specific primers, right? Of course, you have to be very crucial when optimizing the PCR conditions. So specificity and amplification efficiency is controlled. What we do after amplification, we go for gel electrophoresis and to purify the PCR products, right? Once the PCR products are purified, then you have to go for sequencing. So the sequencing workflow we know with 16S rRNA sequencing, generally short read lengths to cover specific areas of 16S rRNA gene are done. Entire 16S rRNA is done, but as I have already told you, certain specific regions, maybe in the conserved areas or maybe in the variant domains, depending upon at what stage of classification you are. Initially, maybe 1500 complete, or maybe only the conserved regions first, then the variable regions. So you have to amplify those, and then you have to go for a sequencing. All right. Now, the final phase is going to be quality control and filtering the sequencing data. So that is going to be the inbuilt and very must important step. You have to filter the sequencing data. There shouldn't be any noise, right? So, uh, utilization of 16S rRNA sequencing analysis softwares are available. For example, QIIME, and that is how you can remember it. QIIME, or Mopur is there. There are many such softwares, of course, and many are upgrading also. So new versions are also on their market. So what they do clustering, annotation, and classifying sequences. That is how they work. So when we say all these words, cluster, annotation, classification, we know that amplicon sequences, what you have, there are variations. So taxonomic annotation is to be done and diversity analysis is to be done, right? So mm, amplicon is the one which is operational taxonomic unit that we are referring to. So the outcome can be used to compare microbial compositions from different samples that you have collected. It can be a sample from water sample, natural sample, or it can be a pus sample also, right? For a specific bacteria you want to identify which is not responding to an antibiotic. So overall, we can say 16S rRNA sequencing is referred to as a rapid and high throughput method, which is a culture-free technique, right? And requires minimum tool set as against DNA hybridization, and outcome are more promising. So when it comes to microbial diversity, 16S rRNA sequences are analyzed, and you are going to understand species composition, its abundance and distribution, community structure of a microbial group. I repeat, when 16S rRNA sequencing is used as a tool in study of microbial systematics and diversity, what all you can do is you can identify the sequence and you can find out what the organism it is, right? That is one. Then you can understand the species composition. You can understand how abundant that particular species is in the given environment. So abundance distribution. So not only of one species, but multiple species which are present in that particular sample can be analyzed, and abundance distribution of each can be identified. Then comes community structure. So once you know the types of species present, the types of species which represent that community, what is their abundance, then you can find out or build up the community structure of microbial groups. So overall, you can get to know about the microbial uh, scenario of that environment or niche. So diversity of microbial communities, and you can say in the ecological systems, right? What is the diversity of microbial community in a given ecological system can be very well understood. So when it comes to analysis, as we had talked about earlier, metagenomic research, integration of 16SrRNA sequencing, and other omic methodologies like DNA hybridization, GC content, TM. Are they absolute? No, those are genomic methods, right? So genomic methods, then 16S sequencing, metagenomic research, transcriptomics, metabolomics, all together can be put to understand the structure and functionality of microbial communities, right? So what we call it as integrative approach or polyphasic approach, in which you have 16SrRNA as one of the tools. Yes, it has got major importance considering the benefits and taxonomical highlights, you can say, or advantages, when you are talking about the conserved domains and the variant domains and the evolutionary importance. All these things we had talked in previous lecture, sorry, in the previous slides. So when it comes to 16S rRNA sequencing, here is one of the slides from um, array- yeah, one of the slides from which is available on internet. I'm trying to share with you. Just give me a second. Till then, you can just write down in the chat what are the keywords that we talked about with reference to DNA hybridization, with reference to 16S rRNA, and I'm talking about its application. Yes, I just want the keywords to be typed from your side so that I get to know how much you understood. Yes, nothing is there in the chat box. See, to summarize again, when we talk about the sequences, DNA hybridization, what are the limitations? Earlier, it was said to be as a gold standard in 1980s when for phylogeny, taxonomic classification of bacterial species and isolates. But with the time, it was found that 70% boundary value, that is set by DNA hybridization, right, is just not enough for classification and taxonomic positioning of bacteria. That is what you cannot really classify many of them. There are certain limitations. Yes, 60% to 70% similarity or differences are going to be enough to reveal hundreds of them, but not all of them. You need to have some powerful tools. So you have different approaches. That is first point we have talked about. Then we talked about what is ANI, what is MUM, what is multi-locus analysis, right? These terminologies we discussed. Then now when it comes to 16S rRNA sequence. So you're talking about a particular region in the entire genome that is encoding 16S rRNA, right? Now here the concern is when it comes to any bacterial cell, you have two important things. That is the set of genes that you analyze. They can be classified as housekeeping genes or the core genes, right? That could be one part. And which are the majority of the genes, and in majority of the members of the species they are going to be present. And second group is auxiliary genes, which are present in some of the members. So you have two terms in addition to DDH, in addition to multilocus analysis, in addition to ANI. Now another term, that is auxiliary genes and core genes. So core genes which are present in the majority of the members of a species, whereas auxiliary genes are present in some of the members. So core genes are essentially those which are called, as I said earlier, what are they called? housekeeping genes. And generally, a sequence of such a gene can be recognized uh, as being within or outside the range of well-studied species. Right? But in the case of for those organisms in which such data are not routinely used, they are multi-locus sequence typings, right? So major factor when you talk about species specificity in the core genes is existence of recombination barrier between the species, right? And recombination methods, natural methods, conjugation, transformation, etc. So uh, frequency of homologous recombination when you talk about decreases exponentially the degree of DNA sequence divergence, right? So this is what is very important. Of course, this is what I have typed in one of the slides. I shall share that slide so that you can take a photograph and read it again what I'm talking about, right? So I'll just try to open that slide. Just give me a second. Yes, it is here. Right, it is taken from one of the research papers, so the terminologies are in blue. Right, now you have the housekeeping genes, you have the core genes, you have multi-sequence multi-locus sequence typing. That is what you are going to do. So when you talk about divergence and when you talk about conservance, so rRNA genome is the one which is having all these important features. That's why it is one of the prime gene sequence that is explored. Right, this is the slide what I had uh, um, shared with you. Sorry, not this one, this one. Right, what are the benefits of using 16SrRNA sequencing? Right, and the last point is what are the steps? So 16SrRNA sequencing flowchart I had shown you in the previous diagram. Now here it is, the notes, so that you can use it in writing the answer. So sequencing the PCR products on plasmid vector, aligning them. So this is what is nothing but construction of the library. That is what you do after you extract the DNA, and it is to be cloned, right? So part by part you can sequencing. Now then you have to go for process, that is amplification. So PCR amplification, and then you have to go for amplified sequence is to be uh, uh, understood, analyzed to know what is actual sequence in it, right? So that sequence can be analyzed by conducting RFLP analysis on PCR products, or you can go by uh, uh, enzyme digestion electrophoresis patterns. So many ways you can extract the information about of the libraries, right? But at the same time, NGS based methods, you can extract the sequence, you can extract the sequence of that particular fragment, and then you can analyze the data. That is the last step in the process. But overall, when I say how you are going to approach the method, that 16S rRNA sequencing analysis, right? So cloning, sequencing, enzyme digestion, right? Then hybridization, sequence analysis, and data analysis. So this is how it goes, and these are the major steps in 16S rRNA sequencing. And why are they required? So this is what we had started with. So here I would like to end and stop my discussion. If you have anything to talk on, kindly type in the chat box, or maybe you ask. If possible, I'll try to answer.